All right, hello everyone. So we will begin soon. Our speaker Wolfgang is here. So let's just give a little bit time for people to join and we'll start in a minute. Okay, well, so why don't we go ahead and get started? So welcome everyone to the Allen Institute Distinguished Seminar. Our speaker today is Professor Wolfgang Maass from Graz University of Technology in Austria. Just a quick biographical note. So Wolfgang received his PhD and obtained habilitation both in mathematics from the Ludwig Maximilians Universität in Munich, in Germany. Later, he did postdoctoral work under the Heisenberg Fellowship at MIT, University of Chicago, and at Berkeley. And then he held professorship positions in computer science, first at the University of Illinois in Chicago, and later and currently at the Graz University of Technology, where he also founded and headed the Institute of Theoretical Computer Science. So Wolfgang is, of course, well known for various contributions in the area of computation and learning in networks of spiking neurons, including implementations in neuromorphic chips. And today, he will tell us about some very interesting work that his group has done using the Allen Institute data and network models, which also has led to a recent collaboration with us. So just a quick note before starting. For the audience, please ask questions. So to ask questions, you will need to post them in the Q&A box. Just type them in and I will be moderating and asking questions, reading them for Wolfgang so that he can answer. So we will probably aim to answer most of the questions towards the end. So please feel free to submit them when you feel like it, but then we will read your questions after the main lecture is done, and then there will be some time for Q&A and Wolfgang will answer. All right, so with that, Wolfgang, thank you very much for speaking here today, and we are excited to learn from you.
Thank you, Anton. Yeah, I'm very glad to give this talk because I think now this work that I'm discussing is something which really requires more of a community effort. And one of my goals today is really to find partners, groups who want to participate in this type of work then. So first now, let me give credits. So the primary work was done by Guozhang Chen, who is just now got assistant professor position at Peking University, which is, I think, the most desirable junior position in China. And Franz Scher in earlier years now, who mainly worked on this programming part, and he's now at Huawei Research in Zurich. So this work that I'm discussing is now available. One is in a published paper already last year in Science Advances, and one is a preprint from September. And so this was funded largely by the Human Brain Project of the EU and a bit from Intel Corporation. So I will discuss how one can train a model for patch OV1 to solve visual processing tasks. I will show you how one can analyze organization of computations in this model. Then one finds functional differences to common neural network models. And they will discuss the astounding noise robustness of the V1 model and come to summary and then outlook. So I will focus on a model for patch of mouse V1 from the lab of Anton Arkhipov. It was published in Neuron paper 2020, Billy et al. Then, and also appreciate the help of Anton and his team in using this data now, which are actually public, which is wonderful. And for me, it actually made a dream come true, which I had this dream for several decades already, but we just didn't have the data available for this then. But at the same time, as you will see also, there is now a confluence of know-how from computer science, computing science, which makes this feasible, this work that I'm discussing. So this, we're looking at this core of this patcher V1, which has an 800 micron diameter, consists of 52K neurons. It's partitioned into one class of excitatory and three classes of inhibitor neurons, the standard ones, PV, somatosensory, and VIP-like neurons on 5 different layers, where layer 2, 3 are lumped together in this data. And what's nice also for computation people like us, this comes together with a qualitative model for the retina and LGN, so they can give to the model the same images which are shown in the experiments to the mouse, and then this feeds synaptic currents into the neurons in this V1 model then. Use the point neuron models. We have to use the point neuron models because otherwise we couldn't simulate it efficiently enough. But also passive dendrites will not help much. I will discuss this a bit later. So in this Billy et al. Model, now the point neuron models are generalized leaky integrated fire neurons, which have 3 internal variables. In addition to the membrane potential, they have also inhibitory currents, which are triggered by spikes of the neurons. And so in this way, you can model also adapting neurons or something like spike frequency adaptation. So technically, the group, the authors of this Billy paper, they selected 111 neurons from the Allen Brain Atlas and then fitted this GLIF3 neuron models to this data. One remark on this side, I would actually think it would be good to use instead the spike response model, because in general it has a better fitting performance to concrete data and probably can be simulated at least as efficiently. So then the core of this data are really connection probabilities. So you have for each of these 5 different layers, you see here this 4 different major classes of neuron, source neurons, and here target neurons. You have connection probabilities then, and so this essential data here is indicated with more redder is stronger connectivity. You see there are many still white spots in this, and also you see other areas where the same number occurs in this table several times then. So I hope this table will fill up over the time. So these are what one could call base connection probabilities, dependence on the type of the class of the pre and postsynaptic neuron. But this is then multiplied with a scaling, which is shown here then, which depends on the distance, horizontal distance of the stomata of the pre and postsynaptic neuron. So it decays within roughly 200 microns in this model then. And we're obviously missing here more detailed connectivity data. I mentioned this 111 types of neurons. It would be nice to have for every pair of them also connection probabilities. So we don't have this for now then. So the data, the weights were chosen already in this model from the Allen Institute, largely random, but with some tuning to provide a receptive fields, orientation, selectivity, and so as in biological data. But it was not really made to carry out network computations. So one remark also about noise. There was a simple noise model provided in this model from Billy et al. Then we applied it if we found that this model was both too weak compared with biological data, but also it had a small artifact synchronization then. And so we replaced it by a stronger noise model that we fitted to experimental data, which were given in String et al. In the Nature paper a few years ago. And you see here on the right these are the same neuron, but 10 or 12 different trials, and you see here there's a substantial trial-to-trial variability. Here it's the same for another neuron then. So not just the spike time is moved, but also sometimes spikes are deleted or inserted. So then we trained this model as it was published for 5 different visual processing tasks. One of these tasks was an artificial task, namely classification of handwritten digits. Apparently mice were never trained to do this, but no, the other 4 tasks were tasks for which mice were actually trained, like fine orientation discrimination in Romancev paper, then visual change detection, which is used a lot now in the Allen Institute in vivo data, where the mouse gets to see a sequence of images with a delay in between then, and in the delay, the mouse has to respond if it was a different image than this one seen before. The same for oriented gratings. And finally, the evidence accumulation tasks, which was developed in the Tank Lab in virtual reality, is one task where the mouse runs down a corridor in a virtual reality. It sees cues on the left and the right. And when it comes to the T-junction, it has to go to that side on which it saw most of the cues. So the mouse has to count, compare numbers, and then remember the result and make a decision on this basis. So we trained this model now for all 5 tasks simultaneously intermixed then. One important design decision we made, and everybody has to make, is really how do you actually want that this network reports the result of a network computation. And we try to do this as close as possible to biological reality, which shows for each possible network decision a small population of neurons on layer 5, excitatory neurons. And then this population of neurons was supposed to fire, for example, if the image had changed then, and otherwise not to fire then. And this is a important step then, because it deviates from the common practice in computational neuroscience. In almost all neural network models for computation in computational neuroscience that I have seen, one takes a so-called linear readout then, which is a neuron, a hypothetical neuron, which gets input from all the neurons in this network, and then you train the weights of this linear readout then. So this looks innocent enough, but there are kind of results which show that, first, let me say, these global readout neurons, of course, don't exist, but also there is something methodological which is problematic, because even without training anything in the network, by just training the readout weights, you can make this whole arrangement solve really complicated tasks, like solve all these 5 tasks without ever not telling these neurons inside the circuit what task it was supposed to solve. So this is simply a result which one knows from machine learning. If you have many variations of some features presented in a recurrent network, then a linear readout has really very powerful classification results on any information contained in this pool of neurons. So here in this design decision that we used for reporting the result of a computation, you see then really the network has to make a particular population of neurons fire at the right time, and these neurons have the same connectivity as any other neurons then. And I think this is looking at the computational task, at making a network decision more from the perspective of this actually circuitry in the brain then. Just one technical slide. We trained this network through some suitable approximation of stochastic gradient descent or backprop through time then. I don't want to say this is the way how it happens in the brain. This is how we did it now to make this network perform computation. And you see in the top line of the loss function has several terms. One is the cross entropy, simply to solving this task, then make this right kind of projection neurons fire at the right time then. Then there is a rate regularization term then here, which makes sure that the neurons in general fire at rates which is fits to biological data. And then there is the last term is a more technical one. This membrane potential regularization is needed in order to avoid that this many neurons are pushed too much in the hyperpolarized regime then. So this is now a technical issue then, but it makes a quite difference in the performance. And then also, as you know, one needs to know a zero gradient to do gradient descent because spikes are not differentiable. So one simply smoothes that. And actually, I think now the form of the smoothing has quite a bit of impact on the result that you get. And so this altogether now, what also turned out to work very well, we coded the whole model, now all these 50 take-home neurons, 111 different neuron types in TensorFlow, and then it can be simulated even on a single GPU then, and even quite fast, then it just takes 3 times more than biological time. And if you also compute here, this backprop gradients, it takes 8 times longer than biological time, which is still okay then. One needs a lot of compute time because you have to train the network on many different examples. We will see this later. And we use 160 GPUs now in parallel to speed up the training, then different GPUs now looking at different training data. And so one now looks what was the outcome of this training. One sees, well, let me first say, we never changed synaptic connectivity. This is, of course, a biological area. No synaptic connections do change. One can add this in later versions of the model then. If one looks at the impact of this training on the weights, one sees that in general, excitatory weights were reduced and inhibitory weights were decreased. And if one looks here now in this plot at the bottom, you see this distribution of firing rates after training is now the dark blue curve, and the dotted line, the background, are experimental data. And one sees that and here the light blue curve are the distribution of rates before the training. So one sees that actually the training moved this rate distribution closer to the recorded data. And this is interesting from the perspective of simply computation science, because the mean firing rate are around 4 Hertz. And usually now in the hands of computer science and neuromorphic engineers, you work with much higher firing rates, and you pay for this by not having really an energy efficient computation. So there were 2 nice surprises also, that actually here now on this plot, we have now we have plotted for the training for several epochs. So 6 epochs take 24 hours of training on 160 GPU for one of these models. And so here's now the learning curve of the V1 model in blue at the top then. And you see it actually learned faster than any of the other control models. And there are several control modes of interest. Here, for example, here this yellow one is of interest because it's a variant of the V1 model where we destroyed the laminar structure then, or simply all spatial structure. We kept the same neurons, but we randomly connected the neurons instead of using this database connection probabilities, which has a strong bias to nearby connections then. And we see now that this has actually a strong impact on the learning speed and even to the performance which is reached at the end. And we have also theoretical explanation to this then, but it's maybe not so important. Here we also looked which is interesting, at some models now which are more commonly used than in computational neuroscience, like really RSNN is simply a recurrent network of spiky neurons randomly connected. All these models here have the same number of neurons, the same number of synaptic connections. And you see here, this green curve also now stays below here, this V1 model. So we see here that this extra biological data about this V1 model have captured some insight into computing and coding, which a generic recurrent network of spiky neurons does not have then. And so here now at the bottom, I have wrote down another surprise, that this V1 model did not overfit. In general, now if one trains a very large model like this V1 model on relatively simple data, you would now expect to overfit. And the contrary was actually the case. Then I will discuss this later a bit. So then we wanted to understand now, how does this network actually solve this task then? And so we homed in on 2 of these 5 tasks, one of the visual change detection tasks, which was also used in wonderful work from the Allen Institute in vivo data as the test on the mouse then. And so here the paradigm, as I mentioned now, is show sequences of images, natural images are taken from ImageNet with some delay in between. Then we had to choose actually in our simulation a delay of only 200 milliseconds between images, whereas in the mouse experiments, these were 500 milliseconds. So this comes to some extent because in this method that we're using, then we have trouble simulating long trials then, and so therefore these are 600 millisecond trials, were in principle good then. But there's also, I think, now as far as we could see, this V1 model on its own could not solve this task well if one has larger delays. So one of our hypotheses is that the reason why the mouse uses also other parts of the brain for solving this task is to make it work this computation for larger delays between images. And this we trained on 40 training images, and all the testing data, all the data that I show you are for new images, which this network never had seen during training then. And so here's homing into the timing for a single image presentation. So these are 100 milliseconds. And here at the top, you see the output of this LGN model from Billy et al. Then. So one sees it mainly becomes active about 50 milliseconds after image onset. And I will later discuss this particular period of 50 to 100 milliseconds after image onset, because this is when really the information about the image gets into the network, and as we see later, also computation sets in. We see activity sets in on layer 4 then particularly. And here now, shown here now, this is the time window where this projection neurons were trained to fire if there was a change. So in this case was a change because this image now has a different color than this image. And so you see, this is here the plot of the spike raster of the projection neurons. And this did come on and fire then for this period. So here now we showed an experiment with many readout of projection neurons. One could just as well take 1 or 2. We never saw any impact on the result of this. Also, it didn't matter whether these projection neurons were chosen close together or spread out over layer 5 then. So there seems to be some general properties which don't depend on these details of the experiment. So this is one analysis that I like very much. Then one had seen the similar work also now in C. elegans from the Simmer lab some while ago, that one really looks at the full dynamics of this network computation then. And so this is made possible by first taking the 1,500 most important PCA components of this 52K dimensional vectors, which record for each 20 millisecond time bin where there was 0 spike, 1 spike or 2 spikes. Then from this 1,500 dimensional data, we projected by UMAP into 2 dimension, and UMAP tries to preserve distances between data points if they're not too far away from each other. And so this is here now an outcome here. So you see there is kind of a middle region, and then when you see here this kind of protrusions, these are each for one of these test images which were shown then. And furthermore, you see even more than that there is one kind of branch which is colored light blue, indicating that these were bundles of trajectories for presentations where no change took place, and here where a change took place than the other one. So this hard formed bundles arise when during this relevant period of 50 to 100 milliseconds, you have a lot of network activity. So this is this very fast moving out. You don't see the trajectory. Here, the main part of the computation takes place, change or no change. And depending on this, you see this bifurcation of this trajectory of the network dynamics. And you see here now, obviously, you have a separate, initially, a bifurcation for each image, which occurs then because this network dynamics moves to a different region then. But then, the second bifurcation occurs then here, as I've shown already here, then depending on change or no change. And so here's a blow up of this particular one then. So we have here current image 7 is shown then, this dog face, and green was the case, that the preceding image was different. So it goes to the change condition, and you see here here is this second bifurcation, which takes place then. Here the time is put on. So this is the image onset. This is 50 milliseconds after image onset. This is 100 milliseconds after image onset. And then the bifurcation becomes visible. And this is then here the time when the projection neurons actually have to become active then. So therefore, now this is our understanding now of how this network computes. Now it has a system of bifurcations, but also we can in the model also analyze what are the neural mechanisms that cause these bifurcations, and particularly the second bifurcation is obviously the more interesting one. And so we could look at this and do manipulations and measurements, which are probably for wet lab neuroscientists, not dream experiments. And we can do this now also in this computer model already. So we analyzed, for example, for each neuron, for each time when its mutual information expressed through its current firing activity with the upcoming network decision then. And we plotted here in this plot on the right. So this is the image presentation. So here during this first period, hardly anybody knows what is the image. Here, this first information about the image is in the network. And you see here some weakly colored spots then here seen from the top, here from the side. So mainly in layer 4 and layer 2, 3 then. These are the ones which have already information about the upcoming network decision. And the saturation of the color corresponds to the mutual information. This is here in this case, the location of the projection neuron, which has to report the result then. And so it's interesting also, to look at those among these neurons here, which had the largest mutual information. Where are they? So there we see here, as I mentioned before, mostly layer 4, layer 2, 3. Interesting is actually that all types of neurons occurred in this then. If one takes a closer look, so not just pyramidal cells then. So this is, I think, a hypothesis which should be tested in vivo then. And also one can in this model now also look inside the neurons. One can ask, how do these early informer neurons, as we call them, how can they have so early an inkling that there is now a change or no change condition? And so this comes because they have internal processes. I mentioned this slow inhibitory currents, which is caused by each spike of them. And if one looks at the values of this after spike currents, then inside the neurons, at the beginning of the next image, one sees that the current value of this current is informative about the preceding image, which was shown some 100 milliseconds ago then. You see this neuron here has a particular strong value of this current. If 6 was the label of the preceding image, this one for if 5 was a preceding image. So you see here then in many different ways do the neurons are able to provide information about the image which is no longer in front of them. And what this is, I think, a remarkable process, because as I mentioned, we are testing this here and doing this analysis for images which the network had never seen during training. So therefore, we conclude now that neurons have really learned to extract generic features from sequences of neurons of images, which enable them to decide whether it was a change or no change condition then. And one can also now, obviously the next question is, are these neurons which have this early information also causally related to the network decision? And so this is easy now to test in a model. We can simply silence the one which have the 100 with the largest mutual information. And one sees then doing this, on one hand, you do flip then here this trajectory from change condition to change condition then. So the whole network dynamics is affected. You can also look at the firing response of the neuron then. So this is here the intact model here showing in the top row, and this neuron fired now for the change condition here shown on the left as it was supposed to. But then if we, after this manipulation, when we silence this 100 neurons in this trial, it was now close to the firing threshold, and another trial, it was now below the threshold. We saw even now that silencing of this neuron also caused hyperpolarization of the model of this projection neuron when it was supposed not to fire. Then you see here in the intact model, it was strongly hyperpolarized. It was less hyperpolarized when these 100 early informer neurons were silenced. Then, so one sees here, sorry, other way around. So here no one sees that if we silence these neurons here now, then this hyperpolarization is also reduced then of these neurons at the right time then. So we want to make sure now that this structural analysis was not just valid for the single task, the change detection task. So we also now carry out the same analysis for this evidence accumulation task that I mentioned before then. And we looked also only at the case where 4 different cues were presented. No, actually, no, sorry, 5 different cues, but we looked here, formed in the case that always the first cue was left, the second was right, the third one was left. And then we only looked at these 4 cases of this last 2 cues then here, shown here then. And you see again here, now this is now the bundles of all these trajectories. This is the first bifurcation for this, the third cue then, identity of the third cue or the site of the third cue. This is for the 4th cue, and this is then for the 5th cue. And you see here then, now one can also now here after the 3rd bifurcation come back to this left turn condition, and you see here this merging of this bundle of trajectories, which was then first now aiming at now turn right, but then now seeing this last cue then on the left, then it was actually merged into this bundle, which here moved then to the turn left then. So we propose now that this analysis of bifurcations will be of general interest, and it would be very nice to do the same analysis in vivo. This is hopefully possible then. And also now carry out this analysis, what causes these bifurcations. And here one can show again that this relatively small population of neurons that are pivotal for this bifurcation, meaning then if we silence them, this bifurcation no longer takes properly place then. So this early informer neurons, or these neurons which cause these bifurcations, then can also be seen in a larger context. So in a very old model from the 90s, Tononi, Spons, and Edelman had proposed just looking at general properties of brain networks and how they possibly compute and how one can analyze what makes them really suitable for computation. They proposed to look at them both at their functional segregation, meaning that different parts of the brain specialized for different tasks, for different types of information. But in the end, you have also integration of information so that the whole brain can arrive at a coherent view then. And you can view this kind of early informer neurons that I have shown you in this examples and the model as an example of segregation then, because these are specialized for controlling a particular bifurcation in the course of a computation then. And the segregation was also now found later now on the level of neurons. So there's the work of Haun and Bresch, 2008, where even exciting a single neurons can cause a change in the perception or behavior of an animal, this manipulation artificially exciting some neurons is a bit problematic because you don't know how many other neurons are excited through this then. So therefore, we prefer to use this experiment of silencing neurons because this hopefully had fewer kind of unmeasurable side effects then. And what's interesting, because I think also having this small set of neurons whose deletion can really abort the computation capability of the network for a particular type of computation, we propose that this is characteristic for this V1 model from the Allen Institute. And this is something that we have not seen in any artificial neural network model, spiking or non-spiking, that we also consider them. So here now to show some data here now in this top plot is here shown the result when you silence 100 neurons, early informer neurons. Here also is information on those neurons which have long time constant and short time constant. So one sees the ones with long time constant are even better in killing the performance when you silence them. Here you see as control if you simply randomly silence the same number of neurons here shown on the x-axis. Here now below is a control for recurrent artificial neural networks randomly connected, the same size. So this has been proposed in several articles in recent years in Neuron and other top journals as being the suitable model for neural networks of the neocortex. And here you see their behavior is totally different than in order to, so here now again is shown the silencing experiment for the V1 model. In order to bring it down to this level, even if you silence in this recurrent artificial neural network thousands of neurons, you never bring down the performance so much. So I have the impression that these randomly connected neurons simply compute and deal with information about the input in very different ways, where the information is a bit everywhere, nowhere, whereas the V1 model appears to be both according to the model and the data, one where you have more specialized neurons. It has a little bit more like a CPU-like precision, where you have a division of labor, and circuit modules have particular tasks then in this whole computation. And I think, therefore, we do need to study more this database model, because these only allow us to understand how these computations are really organized then. So I quickly also wanted to mention another characteristic feature of cortical computation, which the V1 model reproduces nicely, but which we could not reproduce in artificial neural networks. And so this is this combination of sparse sequential firing activity together with what one would call soft rank order coding, namely information in the order when a neuron fires. So we used here this standard paradigm, which was introduced, I think, in Driscoll 2017 from the Harvey lab, and then later from the Tank lab last year in Koa et al. So here you see applied to this change detection task, then you order the neurons in each layer according to their preferred peak activity. So you first for each neuron, your average is activity over 200 trials. Then you compute when is its peak activity, and then you order the neurons, say, on layer 4 according to this peak activity. And you see here there's a certain sequence of activity in layer 4, even somewhat more pronounced in layer 2 or 3 then. And this order depends on the image then. So if you keep this ordering of the neurons here from here then, which shows this nice sequence for image 1, and show another image, you no longer see any sequence here then. And so this you could view as a soft rank order coding or temporal coding, which has been suspected for a long time to be one really of the powers of spiking neural networks, that they compute with time. And one sees here a bit a biological realization of this then. But I call it soft rank order coding, as opposed to this more theoretically studied rank order coding of single spikes, because here we're talking here about firing rates then, right? And you see also smeared out activity. The neuron fires not just only here, but also afterwards, a bit before then. And so we also know in the model we could analyze what is information content of this rank order of firing. And we found now that it contains information not only about the current image, but also the previous image, and also about whether it's a change or no change condition then. And this was not yet had been found or analyzed in vivo then, whether this rank order contains information about the current and preceding image. And so therefore, Guozhang Chen then analyzed neuropixel data, public data from the Allen Institute, and verified that also there the rank order does encode information about the currently shown image and also about the previous image. And I mentioned this as a nice kind of step here now, which is relatively easy when one works with a stutter-based model, because moving between the model and in vivo data is easy because both have the same objects, different neuron types and different locations in the circuit, and they speak the same language then. And I think this is really the power of this type of modeling approach, that models don't live in a world on their own, they live in the same world as the experimental data. So here is an attempt to analyze the same rank order coding in artificial neural network, where the data are prepared in the same way, that for each neuron we normalize its activity over time. And you see most neurons, their fire actually prefer to fire at a relatively long period, maybe 50 to 100 milliseconds. And also at any point in time, you see that actually quite a number of neurons are always active then, right? So one sees here again that this recurrent artificial neural networks has a really a different strategy, which is more like a mass movement rather than this refined almost Swiss clock-like computation that we find in the V1 model. So we also know then, this is in our recent bioRxiv report, we quantified this type of properties, which we found as characteristic properties of in vivo data, which can be reproduced in the V1 model, but are not in any other neural network model that we could find then. On one hand, this is Kandel tau measures this capability of rank order coding. And so close to 0 means here good rank order coding. And one sees here, this had been recorded for the mouse. Now it's very close to 0. It's almost the same value for the V1 model. Whereas if you move to recurrent artificial neural networks, it's 2 orders of magnitude higher. And if you carry out manipulations on this V1 model, say if you remove its neuron diversity or remove its laminar structure, it's already by 1 order of magnitude worse here then. So one sees here that both of these characteristic aspects of the V1 model really contribute to its specific type of computation then. We have a similar analysis here for this function segregation then, where again we see then on one hand we have the V1 model, we have then on the other end artificial neural networks, which have a quite different properties because you don't see this fine function segregation. And if you remove particular parts of the V1 models, you get values somewhere in between there. So then the final point I want to mention is another small surprise, this which I mentioned at the beginning, this astounding noise robustness then. So we have chosen here, as I mentioned, we try to fit the noise to biological data. And so now one can still has a parameter, the noise amplitude. So the amplitude 2 seems to fit well to biological data. When one now chooses a much larger amplitude, like 10 times as large, one sees the performance of the V1 model is still very high then. But if you have, for example, a V1 model where you remove the laminar structure, you have already lost lots of the performance. And if you move to a recurrent network of spiking neurons, which is here this green line, you also have lost quite a bit of performance. So we see here this blue line, this V1 model, is really being amazingly robust then if we make the noise larger and larger. And one should say, no, we never trained this model on noise then. So this holds, the same holds also if during training we applied zero noise then. So this is really a kind of the opposite of overfitting, that there's a free generalization taking place in the V1 model, that it is robust, its computations are robust to types of noise which it had never seen before. Let me quickly also show you something similar which takes place if instead we put the noise into the visual input, we put pixel noise into the pixels for this MNIST classification. And so if we increase the noise amplitude, we see here the performance of this V1 model stays high, whereas convolutional networks, which are the state-of-the-art neural networks in AI for image processing, they're kind of break down then at this point. So then the question is also, why is this V1 model so much more noise robust? And so we have 4 different answers. Now, giving the time, I want to just very quickly go through them. The most mechanistic answer is simply when you look at the responses of neurons in the V1 model to this noise in the input, you see the standard deviation of the neural firing is still almost 0, whereas for a recurrent convolutional network, it goes up first in layer 1, then but also in the other layers. So I conjecture that this is simply a result of having in the V1 model neurons which get many more presynaptic input from many more presynaptic neurons than as opposed to recurrent convolutional networks. So this is a mechanistic explanation. There's an explanation in terms of criticality, because this V1 model operates in a slightly subcritical regime then, and training moves them even closer to the subcritical regime, actually to the same criticality regime as has been reported for red data. There is a more sophisticated analysis of the Stringer theory, where one looked at the power of the variance captured by PC dimension with the log scale. So there's a power decay, and the alpha here is interesting here because the Stringer theory computed an optimal value for this alpha, where D is the number of images in this case then. And one sees that the brain actually, this is the data from Stringer, is very close to this optimal value 1 + 2 over D then. And if one moves higher, then one has more robustness but loses information. Here one has more information about input but lose robustness. And you see here both recurrent and feed-forward convolutional networks are in this region. So therefore, they're really model types which live in this world where coding precision is important, not so much robustness, whereas these biological models live in this world. And one sees again, this training moves here, this V1 model closer to this ideal line here then. And then there's also the Rymantsev theory, where one can see that the information about images tends to come after training orthogonal to the most important eigenvectors of the noise. So my summary is it's now feasible to train large database model for V1, but also you can do the same thing for other brain areas to carry out the same computations or behavioral task as in vivo. And now this provides us some hypothesis about how also network computations are organized in V1, then namely organized through bifurcations, for which small sets of neurons cause these bifurcations. And now these predictions will hopefully be tested in vivo. This pivotal neuron tend to benefit from having an internal dynamics with large time constants, which is probably also one source of deviations between the V1 model and other non-neurons models, artificial neural network models, or standard spiking models that had been used for modeling. Then there is this astounding robustness to external and extrinsic noise. And I think we have to rethink how we want to model networks of neurons in the neocortex. I think we have to move to these more data-based models, because if you take a closer look, you see that all at least all models I know, spiking or non-spiking, that have previously been used, I can't really reproduce what in vivo data show you. So I have here now also an Outlook slide, if it comes on. Yeah. So networks step. So the next step, we're starting already now to replace this backprop training through experimentally found route for synaptic plasticity. And particular valuable is there, for example, the synaptic plasticity rules, which Jeremiah Cohen, at the Allen Institute has co-authored. Then we also it's now begs to be done to also train also models for cortical microcircuits from non-human primates and humans, because in the computer models we can easily mix and match aspects of this model. We can find out what makes possibly the circuits from monkeys and humans more powerful than one has to now move towards training models for combinations of brain areas. I think particularly valuable would be a combination of a sensory area like V1 with the M1, and Antin Akapov's lab works already on a combined model of V1 and a higher visual area LM. We need to then also bring in some models for active dendrites, because top-down input in V1 arrives typically on apical dendrites and distal dendrites often, and we need to bring this into the model. And we're thinking about what is the simplest model which can produce the functionality. And as I mentioned at the beginning, I think this work really needs collaborations, and we need to build a community for this type of work. So please let me know if you're interested in this. And also, I think this work suggests new directions for research in theoretical neuroscience, and add questions, and it adds question marks to some traditional directions in modeling and theory. I mentioned traditional models are somewhat questioned, but also there's been so much work about EI balance, which in this V1 model never has been issued. So here we can now move to the issue, how do these models actually function? How do brain circuits function? So this is all. Thank you all for your patience.
Great, thank you very much, Wolfgang. Appreciate the presentation. It was wonderful. All right, so we have a little bit of time for questions. I see a few here, and if people have additional questions, please continue posting them in Q&A. All right, so let's start. So first, question from Karel. What exactly was missing before Bille et al.? What is still missing?
I think mostly this connection data between these main types of neurons, then which had these connection probabilities. I think this contains most of the valuable information then. And I haven't seen a similar complete data set for other neurons. So for M1, there is a recent paper by Lytton has done this. I think we need also have these connection probabilities between different brain areas then. But also, of course, I think here, as you all know, one is never at the end, right? These are all heuristic beginnings. Then we have to kind of get a sense what type of data provide what type of new information then. And I would advise against simply adding data because they're around. Then I think we need to kind of get a sense for what types of anatomical, physiological, behavioral data are salient then for this question that at least I'm asking here how do these brain networks function?
Okay, yeah, thank you. So next question from Tom Chartrand. Have you explored to what extent the cell type-wise fitted intrinsic properties of the units are contributing to learning or performance in the network?
Yeah, to performance, I've shown this somewhat. So if one only takes neurons with short time constants, say up to 50 milliseconds, the computing capability for these types of tasks has significantly reduced. Then I think but I think we have not yet fully captured this dynamics of different neurons, because I think also this intrinsic dynamics of neurons has to be complemented by short-term plasticity, which is a very powerful effect, which also goes into hundreds of milliseconds. So it will be affect computations then. And I think we need to have a closer look at this. Then so far, I think we have only brought in a very superficial aspect of this diversity of neuron types. So there was one control condition: if we replace all excited neuron types by a single type and all inhibitory by a single type dynamically, then we do find a loss of performance. But I do think that this actual neural circuits form genetically designed modules, computational modules, and we have to find out what they're doing. And this recent Cambayola paper, for example, focused on this network of VIP neurons and other types of inhibitor neurons, how they interact with pyramidal cells. And I think this is a very good starting point for getting closer to the inner workings of the neurons and the modules.
Great, thank you, Wolfgang. So next question is from Eric Brown. It's interesting that the RSSN models trained poorly on task in terms of both accuracy and speed. What do you think is most important thing they are lacking, e.g. laminar structure versus heterogeneous single neuron dynamics?
Yeah, I think the most important aspect is that simply this random connectivity is, I think, can be a curse for network computation, because if you have anywhere gathered some information in some neuron then in the network, in the next step, this information is spread out all over the network again, because it's randomly connected not to all other neurons then. Whereas this V1 model can form pockets of information or specialization then. And I think this is for me a key difference. Another one is this shorter time constants typically just generic spike neural networks have then.
Okay, thank you. All right, I think we have time for a couple more questions. So one from Stefan Michalas. For that comparison with RNNs, did you use connectivity structure which was similar in statistics with our V1 model?
No, this is not we do. We use the always randomly connected artificial neural networks. So you're suggesting to simply just keep the connectivity structure and replace the neurons by artificial neurons then, right? And one would have to then think about what to do with excited and inhibitory. I think this may become informative then. But my guess is that one still stays at quite a distance of this biological form computation, because one aspect that this recurrent artificial neural networks is lacking is they don't have this firing rate adaptation then, and so one sees then they keep active for a long time, whereas the neurons in V1 model, they fire strongly and then they shut up then, and they're very hard to recruit afterwards. And this, I think, creates this much more temporally precise activity slots for different neurons at different time points in the V1 model.
Great. All right. Question from Stephen Smith. How sensitive are the learning results you described to the regularization parameters used to set up the models prior to training?
I think what we did prior to training did not matter too much then. I forgot, I think maybe you're asking about the initialization. So we initialize typically with Pille et al. So this model, which as it was published, then I think we did some control conditions. If you start out with a more purely random weights, then you need longer to train it then. But I think it's an important question. I also want to mention maybe in this context, one type of work which we briefly started, but where more work should be done, is one sees that simply looking at this connectivity table between different populations of neurons, one can actually use such tables to program computation capabilities into the network without any learning then or training. Then we have shown this separately then. And I think this is probably the way how actually innate computing capabilities are arising in many brain areas, then simply through this connection probability. And key is that for that is that you don't have just 1 or 2 types of neurons, you need at least 10 types of neurons, then only then this connection probability table can inject enough information into the network to make it compute in a certain way.
Great. All right. So maybe one last question, Wolfgang, and then we'll wrap up. So from Eric Brown, one more question. Please tell us more about the way that noise was used here. Was there intrinsic noise present during the BPTT training? And then you later tested in the presence of gradually increasing noise.
Yeah. So as I mentioned, we found then the same robustness result as shown. They also come out when there was no noise during training then. So this was really a free gift then in the end then. And then when we trained it later with increasing noise then, so this was only during testing then, right? So during training, we never had any noise then, or also as a control with the standard noise or so. So only during testing, we increased the noise then.
Well, great. Thank you so much, Wolfgang. This is wonderful. And thanks everyone for joining us today. That concludes our seminar. And Wolfgang, you have several meetings with a number of people from the Institute scheduled for the next few days.
Yeah. And let me maybe also thank you and the other researchers at the Allen Institute to do this really wonderful focused work. I think this is exactly what we need to make progress also on these big theoretical questions. And I'm really glad that the Allen Institute works in this way.
Wonderful. Yes, I agree. Thank you so much. All right. Great.